Now the topic before us is movement of substances in and out of the cell. Some substances like carbon dioxide, which is a cellular waste, requires to be excreted out by the cell. Suppose this is the cell and carbon dioxide is in high concentration. Now we can see that we know that carbon dioxide cannot be seen. For the purpose of example, we are taking this, assuming this. And suppose carbon dioxide is in high concentration inside the cell, and outside the cell the concentration of carbon dioxide is less. As soon as there is difference in concentration of carbon dioxide inside and outside a cell, carbon dioxide moves out of the cell through the cell membrane. This is the cell membrane by the process of diffusion, that is from the area of high concentration to the area of low concentration. Similarly, we again assume that this is the cell and the level of oxygen inside the cell decreases as compared to the concentration of oxygen outside the cell. The oxygen enters the cell through cell membrane by the process of diffusion. Next, water moves in and out of the cell by the process of osmosis. These substances move in and out of the cell from the region of high concentration to the region of low concentration. It means that if the concentration is high in a particular area, it moves from that to the area where there is low concentration of the substance. Now we will understand the difference between hypertonic, hypotonic and isotonic solution on the basis of concentration of solute in the solution. Solutions are of three types: hypertonic solution, hypotonic solution and isotonic solution. We will understand the meaning of solute and solvent in the coming slide. Using these terms, that is hypertonic, hypotonic, isotonic, always involves comparison between two different solutions. Let us assume otherwise. Suppose we say that he is taller. How much taller? He is taller than him. That is, we are making comparison of two persons. Next, if we say that he is more beautiful than her, we are making again comparison of two persons. In the same way, when we are talking about hypertonic solution or hypotonic solution, there must be the presence of some other solution with whom the comparison has to be made. Cells have a very limited range of conditions in which they can survive. It is not that if the conditions are too hot, too cold, too watery, too salty, too acidic or too basic, the cells cannot perform in all these conditions. They can function properly if there is the condition which is suitable to them. So we found out what happens when you place the cell in a solution which is more salty, that is hypertonic, much more watery, that is hypotonic, or has an equal salt concentration, that is isotonic. So we will try to find out the position of the plant cell when it is placed in hypertonic, hypotonic or isotonic solution. Now we will first understand the meaning of solvent and solute. Solutions are mixture of composed of solute and solvent. Suppose this is a jar, this is water. We have put some salt in it. So salt is a solute and water is solvent. A solvent is a substance that dissolves a solute resulting in a solution. A solvent is usually a liquid. Now what about solute? Solute is a substance which is dissolved in another substance. In the example you have seen that the salt has been dissolved in water. Water is a mixture, it is a solution, mixed solution which is composed of water and salt, in which salt is a solute and water is the solvent in which salt or solute has been mixed. Now we will define or understand the meaning of hypertonic, hypotonic and isotonic solution. We will try to understand the meaning with the help of pictures so that we can easily understand the definitions. First, hypertonic solution. In this picture, you can see that these dots are solutes. These are also solutes. So, we are talking about hypertonic solution. The solution which is inside this, this black color covering is hypertonic solution. It has more solute in it as compared to another solution. That is, if we assume that this is the cell and the cell has less solute, whereas inside the cell solute is more. So the solution which is outside the cell is hypertonic solution. So next, hypotonic solution. This is the hypotonic solution in which the cell has been placed. You can see that solute is in low concentration as compared to the another solution, that is inside the solution, inside the cell solutes are in high concentration, whereas outside the cell solutes are in low concentration. So a hypotonic solution is the solution in which solute concentration is low as compared with another solution. Another solution means solution inside the cell. So now talk about the isotonic solution. If we assume that this is the cell and you find that the number of solutes inside the cell and the number of solutes outside the cell are equal. So outside the cell, this is the isotonic solution in which it has the same amount of solutes as compared with another solution, that is solution inside the cell. The number of solutes which are present in the solution outside the cell as well as within the cell are same. So whenever we are talking about hypertonic, hypotonic and isotonic solution, we are talking about the solution which is outside the cell. Thanks for watching. If you like our course, please spare some time to give a star rating to our course.